Somani, he is traveling to Kedarnath, and also he will probably out of connectivity. That's what he has conveyed to me. And conveyed his best wishes. Yes, because I was trying, and then he wrote to me that he's continuously out of coverage. So we will start it in another 10 minutes, another 5 minutes for sure. It's 9:58 now. We will start now. I just inform Manisha to start.
Have we posted?
How was your journey from, as in you said some, yeah, sorry.
This link has been posted in APTI Women Leaders First Conclave group. No.
Yes, ma'am. No, we have. Yeah.
So we expect some people to go.
Right.
You were asking something?
So that, yeah, no, that way though we have received around 700 registrations for this particular event. So let's see how we, and so we have made an eligibility of 300 on the Zoom platform. But then there are more participants, and we So has the YouTube streaming been started?
Started.
Okay, I think, ma'am, we should start.
Yes.
Okay.
So, ma'am, please do start.
Yes. Very good morning to one and all. I am Dr. M.D. Kitukle, Principal of P. Vadhwani College of Pharmacy. Welcome you all for the 3 days webinar on Train the Trailer program that has been arranged by APTI's Women's Forum, Maharashtra State Branch, APTI's Women Forum Western Zone in collaboration with Indira College of Pharmacy. welcome Dr. Vandana Patrawale ma'am, who is the chief guest of today's function. Welcome Dr. Somani, Dr. Anuradha Mujumdar, and all the delegates who are present today for the 3 days webinar. This is the inaugural function, and on behalf of APTI Women Maharashtra State, I invite Dr. Anagha to take over the mic. Please, over to Dr. Anagha.
Yeah. Good morning, one and all, and it gives me immense pleasure to be here amongst you. Like on behalf of APTI Maharashtra State Branch Women's Forum and APTI Western Zone Women's Forum and Indira College of Pharmacy, Pune, I welcome you all for this program. I welcome today's chief guest, Dr. Vandana Patrawale ma'am, and today's esteemed speakers, Dr. Parijat and Dr. Aarti for the same. I also extend this warm welcome to all the participants who have joined through Zoom and through Telegram and through YouTube. So in brief about today's program is like why this Train the Trainers program. We have been receiving feedback from many teachers that they feel inhibited or a bit heavy to teach certain subjects, like which they have not otherwise studied or not otherwise practiced at all. So we thought of arranging this program for updating the pharmacy teachers on the latest developments and advancements in the field of pharmaceutical science, technology, and research. This webinar would definitely help them in improving their classroom delivery, scientific writing, and provide insights about conducting meaningful research. So I also am grateful to all the esteemed speakers who have agreed to deliver their sessions and share their knowledge and experience with all of us. Dr. Parijat, Dr. Aarti, Dr. Sunila, Dr. Indupal Kaur, Dr. Ruxana, and Dr. Anuradha Mujumdar herself. So, dear teachers, it is going to be a great learning for you, and make most of it, understand the things, feel free to ask questions whenever given after the sessions, like of course, and give us feedback about what more you want so that we can plan such activities. Being teachers, we have this responsibility of we are the torchbearers and facilitators, where we will facilitate the students. If we are upskilled and we are in tune with the latest trends, we will be in a position to transfer this knowledge to the our budding students, and in short, we will be providing a good raw material to our pharmaceutical industry. So that's the responsibility on us as teachers to continuously upgrade ourselves and come out of our shells, learn new things, and then pass on the knowledge. So with this short small words, I will again welcome everybody. And thank you for joining, everybody. Thanks a lot.
Okay. Thank you so much for your kind address, Anagha ma'am. Now I request Dr. Anuradha Mujumdar, Bombay College of Pharmacy. She's the Associate Professor of Bombay College of Pharmacy and Coordinator of APTI Women's Wing Western Zone. I request her to please address the session. Over to Dr. Anuradha.
Thank you, madam. Thank you. First of all, I would like to congratulate Indira College of Pharmacy for hosting this wonderful 3-day webinar series on Train the Trainer, which was very much required and pertinent, because we are at the threshold of introduction of new education policies. We've seen the slow emergence of Industry 4.0. Greetings to my senior and highly revered Dr. Vandana Patrawale ma'am, Dr. Rakesh Somani, Dr. Anagha Joshi, principal, who has taken the lead for this webinar. Coordinators, I was really impressed that the coordinators, just like the spirit which is being promulgated by Dr. Vandana Patrawale, coordinators are from different parts of Maharashtra, from Yavatmal, from Jalgaon, from Government Polytechnic, Amravati, and our very own Westcop, Mumbai, Chembur, Mumbai. So coordinators, I would like to thank you for your involvement, Dr. Supriya Shidhaye, Dr. M.D. Kitukale, Mrs. Neha Povar, and Jyotsna Saunere. Greetings to my dear colleague, Dr. Madhur Kulkarni. resource person whom I know and I value their knowledge and their skill and understanding. Our very own Dr. Parizad Enchidana was going to speak on AI and ML simulation. I think, as I again repeat, that we are here, our students need to understand about the emergence of Industry 4.0, going beyond automation, plus the use of AI, ML, IoT in technologies and platforms, and in during research. But Aarti Prabhu was always been the proponent and the user of computer-aided designing. She's next teacher. I feel this is a very good prelude before NEP 2020 based syllabus is being declared by PCI. Because NEP 2020 speaks about interdisciplinary reorientation. My dear students, my dear teachers who are part of this webinar, you need to understand and take cognizance of that in the future, higher education, even pharmacy, would become borderless and seamless, with more of blended, more of, because of the advent of ABC, there would be more flexibility, lot more flexibility for the student community. So we teachers need to get ourselves upskilled and trained, reorient ourselves, uncondition and recondition ourselves to the emerging scenario and ecosystem in higher education and research. Thank you very much, Dr. Anika and Dr. Madhur, for giving me the opportunity to speak on behalf of IPA, of APTI, Western Zone Women's Forum, and I would like to thank Dr. Vandana Travale for taking the lead to mobilizing so many events under the APTI Women's Forum umbrella. Thank you very much. All the very best.
Thank you, ma'am. Thank you for your valuable words. Dear delegates, we have eminent personality as chief guest for inaugural function of this 3 days webinar series. I took this opportunity to introduce her. Dear delegates, Dr. Vandana Patravale, she's currently a professor of pharmaceutics at Institute of Chemical Technology, Mumbai, India, and has around 33 years teaching and research experience. She has over 200 referred publications, 13 grand patents, and 22 in pipeline, and 2 trademark registries. She has published 2 books and 25 books chapter with international publishers. Dr. Patravale has been active in teaching, research, service throughout her career. She has awarded with Kukreja Oration Award 2020, APTI's Dr. Manushree Pal Base Pharmaceutical Scientist Award 2019, and Shri Amrut Modi Distinguished Researcher Award 2018, and OPPI's Woman Scientist Award in 2015. Along with this, she also, at her credit, Best Pharmaceutical Scientist Award 2014 and APTI's Best Teacher Award at 2012. She is All India President of Women's Forum of APTI and Vice President of CRS Indian Chapter, Editorial Board Member of DDTR, and Editor CRS IC and APTI Women's Forum newsletter. She has completed Indo-Swiss, Indo-Japan, Indo-UK projects. She is executing all the major grants from Indian government, focusing on product development. She has transferred about 20 technologies to the industries, including drug-eluting stents being marketed in more than 60 countries. With this huge introduction, I would like Dr. Vandana ma'am to address the session. Over to Dr. Vandana ma'am, please.
Thank you so much, Dr. Manisha, for that kind words of introduction. And good morning, dear all fellow colleagues, I must say. Really happy that APTI, under the presidentship of Dr. Milind Umekar, is moving step by step ahead. And we are aligned with the vision of APTI Women Forum Center activities. And first of the step is to disseminate knowledge amongst our fellow colleagues. Network with each of them in every possible way do hand holding with each other and see that we, the women colleagues, at least are not left behind. So I really, really appreciate Dr. Anagha Joshi for taking this step forward and coming up along with the convener Lavina Jain and organizing committee members Dr. Kitkule, Neha, Jyotsna, as well as Dr. Supriya Shide, under the presidentship of Dr. Rakesh Somani, APTI Maharashtra State, to come up with such a program, train the trainers. Already the objectives have been highlighted by Dr. Anagha Joshi. Though we say that it is young teachers who need to be trained, I feel that it is each one of us who needs this training. We need to unlearn at times and relearn many of the new things which are in the knowledge domain. So prime objective of this Train the Trainer program is, of course, to spread the knowledge and imbibe whatever these experts have to give us so that it can further get disseminated to students. Again, when we talk about teachers, teachers are the facilitators. We learn, we discuss, as we said, we imbibe, and then we give that information to students in the form in which we have learned. But there's a subtle difference, we say, between teacher and guru. And these trainers would be gurus for us. I'm not talking about spiritual gurus. I'm talking terms of gurus as mentors, gurus as guides. And I'm so happy that APTI Maharashtra State Branch has selected those mentors who are ever ready to give their time for academic activities. And I can vouch for it because Dr. Parizat Elchidana, being in fact the president of Controllerly Society also, we have interacted very closely with her. And she's very ready, always ready to learn something new and then teach whatever she has learned in a very selfless manner to the teaching fraternity. So it's really great that we have trainers, as Dr. Anuradha mentioned, Dr. Parizat Elchidana, we have Dr. Aarti, we have Dr. Indupal Kaur, who was recently chief guest of first National APTI Women Leadership Conclave. And leaders have to be such who take the team along. You'll be surprised that for entire 1.5 days, rather I can put it as 2 days, chief guest was also along with all the teachers. And that definitely increases the networking opportunities when you have senior leaders amongst you. So we also have Dr. Ruxana. On Thursday, 4th May, we have Dr. Anuradha Mazumdar as well as Dr. Sunila Dhaneshwar, another leader. And I do not have words. And I really thank APTI Maharashtra State for Women Forum for selecting such apt speakers who would be giving us all we need. But one request, that this activity must continue. The Train the Trainer program can continue, dear teachers, only if you know and you come out with the activities you would like us to put. So feel free, use the chat box, use the email IDs of Women Forum and let us know what is it, what you would like to learn, and we promise that we'll execute all those suggestions and take them forward. We have real good support of the center, and no activity, will be left half done. So with this, I would like to wish APTI Maharashtra State Women Forum all the best. They have really initiated and taken the lead in this. And we hope that in coming days, you'll see a lot of activities coming through this forum. Thank you. Enjoy all the sessions, learn from them, and do not stop there. Learn and teach. Thank you so much.
Thank you, ma'am. Thank you very much for your appreciating and inspiring words. Real as many things is a special quote we took from you. We also take from you that this system is going to spread knowledge, and we can imbibe the experts' knowledge to the student so that we can disseminate it to the students, ma'am. Thank you for inspiration. And the most inspiring thing is the chief guest is going to be with us for next 1.5 or 2 days. That is very inspiring. And we feel very free to use this platform to rise up, ma'am. Thank you for your inspiring word. Thank you once again.
Thank you.
So with this, may I declare the inaugural function is over. And now we can switch over to the first session. Dear friends and teachers, today's for today's first session, the speaker, we get a very eminent speaker. And I'm taking this privilege to introduce Dr. Parizat Ilchidana, ma'am. Dr. Parizat is a PhD in pharmaceutical science and qualified and trained as internal auditor for ISO 9000 and quality management system, diplomas in business management and computer science. She has a huge experience of around 37 years in global generic pharmaceutical industry with specialization in research and development, especially of patents, non-infringing products, and process and quality management system. She has expertise in startup and setting up and managing greenfield projects on behalf of foreign investors. She has delivered numerous global FTF and NCE1 submissions, along with successful launches in regulated and semi-regulated market. She has a few patents and research publications to her credit, and has understanding and knowledge of QHD and AI related to the product and process development and analysis also. Throughout industrial career, she remained close to the academics also. It is an honorary visiting, she's an honorary visiting faculty and is on technical advisory board of various colleges in India and Dubai. She is recognized guide for PhD in couple of universities and well sought for examiner of MPharm and PhD thesis. She is invited speaker at various national and international seminar and symposium, and presently she is the president of CRSIC. So we are very lucky to have you, ma'am. With this, I request her to take over the session. Dr. Parizat, ma'am.
Yeah, thank you, Dr. Kitukale. And thanks, Dr. Joshi, for having me over for this wonderful session and allowing me to share some of my thoughts and experiences from industry. So it's a real pleasure to be here today and speaking to all the eminent professors from various colleges. And if I can start by sharing my screen.
You can start, ma'am. You are a co-host, so you can start sharing.
Okay. Is it visible? Is it visible? Is my screen visible?
Not yet.
No.
No, ma'am.
not sure what's going on. Screen share. Yeah, can you see it now?
Yes.
Yes,
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 visible.
ma'am, it is visible.
Okay. I mean, different platforms have different way of handling things. Right. Okay. I'm not able to. Okay, so today I will be speaking to all of you all on artificial intelligence and machine learning with respect to their applications in pharmaceuticals. Now, first and foremost, let's understand, what is artificial intelligence, because different people have different interpretation of this particular terminology. Artificial intelligence is nothing but simulation of human intelligent processes through machines, and that also especially through computer systems. And it is also a branch of computer science which is concerned with building smart machines, building various algorithms, which are capable of performing tasks to support mainly in the decision-making processes based on the voluminous data that has already been generated and it's available to various algorithms for providing us and helping us in the decision making. And this also is based a lot on data analytics. So it is nothing but a combination of artificial intelligence, machine learning, data analytics, all put together. And as we progress through this session, you will be able to understand this in a much, much better manner. Now, coming to my first slide with respect to the introduction, artificial intelligence powered solutions and machine learning technologies, it can empower the healthcare to embrace a data-driven approach and analyze volumes of data to arrive at the right decision. This is a proper technical definition. What I explained to you was in a very, very simple term, right? So how does it help? It helps in predicting what research will translate into clinical trials and further into commercialization. And this is something which is very, very important for industry. And I request that even in academia, if we can put in more and more of AI in a manner whereby which we can translate the research from academia into industry commercialization, it will be helpful for both academia and industry. It helps to transform the drug discovery by accelerating the research and development timeline. You can really compress the research timeline and thereby even save a lot on the cost of development. It also helps in repurposing of drugs, and this was especially seen during COVID-19 pandemic. How do we think we came up with a vaccine in such a fast manner? It was only because lots and lots of data on vaccine manufacture was available, and through artificial intelligence, we could come out with what would be the best suited vaccine. So how does it evolve? How did artificial intelligence evolve? It evolved through the Industrial Revolution. Industrial Revolution 1, 2, 3, we are all aware of. When it reached to Industrial Revolution 4, we went up to cloud computing, AI, big data, Internet of Things, etc., etc. But where is pharma today? Today, pharma is still lagging behind. We have crossed the barrier of AI. AI is now being implemented in a lot of areas in pharmaceutical industry, but we need to still further go into big data, work on Internet of Things, go to robotics, go to cyber-based systems, digital evaluation. It's a still long way for pharma industry. And not just that, pharma has not even touched I4. We are now moving into I5. And I5 is more and more driven towards robots and smart machines. So this is where we need to be aware. we are still talking of I4 and Pharma 4, whereas other industries are moving towards I5, where sustainability, human centricity, and resilience are also very, very key to the various industries. So how can we apply AI at the various stages of pharma development, right from molecule discovery up to marketing, which is with respect to the supply chain management? So under molecule discovery, we have various opportunities available. Next, we go to the preclinical testing, where earlier was done in animals. Now we are doing an organ on chip. We can move on to formulation development, where we have e-lab notebooks, which have come in, followed by quality by design, which is very, very important nowadays, because you don't do formulation development, you do formulation design. And then to add to that, there is a very, very excellent software on DDD Plus, which is helping in to develop dissolution methods, which would be indicative of an excellent IVE simulation. Then coming to clinical trials, they are becoming more and more simple with wearable devices and simulations. And this over here, I'll present an excellent model of Simulations Plus. Coming to process design, earlier process design was more of a tech transfer, and it was being done just on an ad hoc basis. Currently, it has to be done via quality by design approach, where you are not just developing a process, but you are designing the process to meet your end goals. Coming to quality, both QC and QA, there are various systems which are based on AI, which I will be taking you through shortly, very briefly. On manufacturing side, we have ASRS, operation software, smart machines, continuous processing, 3D printing, and much more. Coming to supply chain, which is more of market-based orientation, where it tracks and traces the product right from the time it is manufactured to the time it reaches the hands of the patient. Not just that, there is also backward tracing from the patient up to the company, up to the batch, and up to the ingredients and excipients which have gone into the batch. So this is the extent to which artificial intelligence, the various softwares, the various algorithms, the various machines, the machine learning has helped pharma industry to evolve right from the molecule to the market. Not just that, when we come to the regulatory submissions, the pharmacovigilance, the product lifecycle management, for that as well, there are a lot of softwares available which will help in managing the various requirements, and which I will touch upon very, very briefly in the later part of my talk. So what is it? Is it something of the future, or is it happening now? Friends, the answer is it is happening now. We are designing new drugs with AI. We are designing new delivery systems with AI. We deliver clinical trials within minutes nowadays. Drugs are not tested on real humans or animals, but on virtual models. Robots are helping in manufacturing of medicines as well as their distribution. Artificial intelligence is helping in process optimization, process controls. Batches are being released virtually instead of individuals tracking the data and releasing the batches. Counterfeiting of drugs hopefully will become almost impossible through the track and trace system, which I talked of earlier. Blockchain technology ensures a complete distribution channel is within the control of the company. And futuristically, we are also anticipating 3D printing of medicines for personalized delivery. Doctors have started printing organs for transplantation, and even instead of studying, there is an opportunity of replacing chips in the brain, which are already having all the information and all the study that you would like to do during the course of your tenure. So this is the extent to which we have developed. So coming to the first part, that is molecule discovery. And I won't go much into details of this. This is a slide which is predicting that you identify a disease, isolate the protein. You can have a molecule which is developed in silico for that particular protein treatment, and then you can straight go on to preclinical trials. And under preclinical trials also, you need not actually do in vivo studies. You can do in vitro and come up with ADMET models, which will help you to come up with a molecule in a much faster manner. So I won't go into the discovery of the bulk drug or the API, because I think the next session will take care of it, but just a few screenshots of the various softwares that are available through AI, which can help you develop the molecule. This is background is the picture of a fungus, and against that, how a drug molecule is developed and given via the software. It's not just that this molecule is presented via the software through artificial intelligence, but even actual synthesis of the molecule. What should be the starting material? What should be the in-process parameters? What would be the additional products coming in? What can possibly be the final product coming out? What can be the purity? How can you improve the purity? What could be the impurity profile? All this information and data can be available through the in silico approaches, through the various softwares that one can use. These are some other model screenshots I have taken and presented for molecules that have been developed in-house in our labs, as well as from certain other labs across the world. Apart from developing a new API, the repurposing of drugs which were already developed, it's a big market for drugs as and when they are repurposed. The main example is aspirin. It is a drug which was developed as an NSAID, non-steroidal anti-inflammatory, but today it is being used as a cardiovascular drug which has been prescribed by cardiac physicians to each and every patient which is coming in. Apart from that, there are certain other drugs like hydroxychloroquine and ivermectin, which came up in use during the COVID scenario. You all must be aware they are some of the drugs which were prescribed in a very big manner, and these drugs came up into action only through repurposing. So what is drug repurposing? It is repositioning of a drug which is already developed and known in the market, and repurposing becomes very fast and a quick way of administering it because the complete profile of the drug is known. The toxicity profile is known, the ADMET is known, the kinetics are known, everything is known for this particular molecule. When you are repurposing it for a different indication, all you need to do is work out the new dose that you will have to use for repurposing and the delivery system, whether it will be oral or injectable or transdermal or topical, that needs to be worked out. So it would be a very fast way of coming up with a new drug. Now let's look at preclinical testing in animals. As I said, US FDA or most of the countries in the world are requesting pharma companies to do away with animal studies, to look at certain other methods by which we can determine the toxicity profile, by which we can determine the activity of a particular molecule. Many methods were available. We were using plate techniques. We were using cell culture techniques in order to determine the activity and toxicity of certain drugs that we were developing in our age. But today, what is available? Today, what is available to us is organ on a chip. Apart from that, there are also requirements where you drop the animal studies and you directly do translational work from the molecule to the patient, and the patient in the clinical trials is directly being monitored for the effectiveness and toxicity of the drug. So coming to organ on a chip, what exactly it is, we do away with animal studies and we look at different types of chips which are different for different organs that you are trying to identify and treat or trying to check the toxicity that it may impact. And this is developed by a company called Emulate. They have done a complete presentation to us through our AAPS Indian chapter. If anyone is interested, you can let us know. We'll get in touch with them and they will do additional presentation to you all. What all organs are available with them? They have done liver, kidney, intestine, brain, and lungs. And here, animal models are totally avoided. 2D cell culture techniques are avoided, and we directly go on to the chip. So what happens actually in the chip? In the chip, especially, I will take you through a chip which is developed for brain treatment, wherein in the first compartment, you have the various cells which are there in the brains like neural, microglia, astrocytes, etc. And then you have an in-between porous membrane, and you have a bottom channel, the red one, through which you will pass the drug. Through the membrane, the drug will get absorbed into the various cells in the upper chamber and will tell us the effectiveness, whether it's posing toxicity or whether it's having a beneficiary effect on the various cells. You can also develop these type of structures even for testing out drugs on various cancerous tissues. It's a very, very new and technology which has come up. It's not very popular in India still. Hardly any companies have started using it, but abroad it's being used very frequently and when they are trying to do away with animal studies. Coming to our formulation design. Now, many a times when we are designing formulations, we can predict the various excipients that we would like to use, but how about the various concentration of the excipients that we can use? And those type of excipients and the concentration of excipients will all depend upon what are you looking for in the end product. What is the diso profile that you are looking for in the end product? What is the disintegration that you are looking for in the end product? Earlier, when we were doing formulation development, it was called development. We would develop a product, we would test it, and whatever results we get, those would be the specifications of our product. No, that's not right, because those specifications may not produce the required in vivo effect that is desired. And hence, we have come to a backward working wherein we define what is the in vivo result that we are expecting and accordingly design the formulation in order to achieve that particular desired effect. And in order to do that, we must have a very, very robust dissolution profiling test, which will actually mimic the in vivo release profile of the drug. It should be a bio-relevant dissolution method, not just a physical dissolution method, which is currently what we're using. They are all for just release of the product to the market. They are quality determining dissolution methods, not methods which are required to achieve whatever we are expecting in vivo. Apart from that, I've put in a brief note on the e-laboratory notebooks also, which is once again a software program designed to replace paper in laboratory notebooks. We are looking at moving towards paperless labs. We created our complete quality lab, which was paperless. Now efforts were being made to put in formulation lab next to make it paperless so that no transcription errors happen. There is no data integrity issues because many a times US FDA finds holes into our data that we have generated, especially if it is hard data, if it is paper data, if it is handwritten data, they are refusing to accept. So a lot of effort is being put to make lab data also electronic. So I think quality by design is something which is already being taught in various colleges. It's a part of the syllabus. But apart from that, there are various formulation development softwares also available, which could be helpful in designing a particular product. But finally, it is QBD, which is one needs to put in place in order to have not just a robust product, but even a robust process. So when you're looking at QBD, you are looking at formulation, you are looking at process, you are looking at various equipments and the materials that come in. And in order to achieve all this, there are various parameters that one needs to look at. That is critical quality attributes, critical material attributes. One needs to use design of experiments, critical process parameters. One has to utilize PAT. Operational excellence has to be brought in. So all these parameters and the number of experiments that one needs to carry out in order to achieve quality by design. And apart from that, even the personnel training, who's working on the project, are they trained for all those projects, has to be put in place. And how can you have such a data, such a voluminous data handling by just a computer system or just a paper-based system? In order to do that, we have various softwares which are designed on artificial intelligence and machine learning. One is a design expert, which is not as sophisticated. Design expert allows you to do various factorial designs, but there are other softwares which are more sophisticated, where you can have complete fusion of the various experiments that are carried out, and you get the final results in the form of a 2D plot or a 3D plot, as and when you are having more number of experiments which are carried out. And why this is very important and why regulatory agencies are insisting upon it is, if you make any changes within the design, then these are not reportable changes. You can make those changes and continue manufacturing. Continue manufacturing. Otherwise, I'm not sure how many of you all are aware, any change that is made on the shop floor once the product is submitted and approved, it is a change which is a reportable change. And if it is a reportable change, one has to wait for approval before the change can be implemented. You can proceed with the additional product batches to be manufactured. But if you have worked on this design space and the changes within the design space, it's not a reportable change. You can proceed and implement the change and go ahead with your batch manufacture. And just one experience of mine, our first ever submission to the US FDA on quality by design basis, we got the approval for manufacturing and marketing that product in 9 months flat. This was the fastest approval that a company would have got. Imagine today's day, approval takes anything from 18 to 24 months. And we could get this reduced to almost 1/3 the timeline, and also a huge reduction on the cost. I keep on mentioning about the dissolution method, how important it is. So there is a method available, there is a software available which helps us to develop dissolution method, which is very close to your IVE, in vitro, in vivo evaluation. And those particular methods that we develop are mimicking what is happening to the drug in vivo. So your correlation is very good in vitro and in vivo, and the methods and the media that you are developing are sustainable and will help you to decide on the complete release profile of the drug that we are intending to have. So what happens here? D plus is dose disintegration and dissolution plus. It is an advanced program which will simulate the in vitro dissolution and disintegration of APIs. Once that is done, and you carry out maybe one preliminary pilot bio, or you know the kinetics of the particular drug, the software will tell you what should be the media for dissolution, what should be the speed for the dissolution, what should be the disso profile, that is the rate of release, whether you need a solubilizer like SLS in the medium, or you can do without a solubilizer. And this will do away with our cut and try methods that we normally use in the labs to develop a particular product. this will give us an assured method which can be further scaled up and will give us excellent in vitro in vivo correlation. This particular model is useful for all powders, capsules, tablets, polymer matrix tablets, coated beads, bilayer tablets, delayed release coated tablets, and they will also correlate well with the various USP apparatuses, that is USP4, 4 USP different apparatuses, paddle, basket, flow through cell, that is apparatus 4, even the rotating disc and the new PUNE disc profiler, which USP has come out with. So this is the extent to which your formulation design will become shortened, and you will be able to come up with the best formulation where you are sure you will be able to achieve the required release profile. Coming to briefly on clinical trials, I think everyone knows about wearable devices, and these wearable devices have helped a lot in the current clinical trials that we are carrying out. Earlier for clinical trials, we had to house the patients, or even if the patients are not housed and are allowed to go home, it was very difficult to monitor them. Now with these wearable devices and the connectivity of those wearable devices with the computers which are present within the lab, it's very easy to monitor and track the patient with whatever parameters that we are studying. So imagine a clinical trial which earlier used to take months to recruit patients or recruit human volunteers can now recruit volunteers much easily, because the volunteer does not have to stay back with you for those many days. He's free to go and he's free to do whatever he wants. So they are now more and more open to participating in those clinical trials, and it's all because of wearables devices and the AI behind them. Coming to whatever I was talking about, GastroPlus, where we have in silico determination of various kinetic parameters. You don't have to actually administer the drug to a human volunteer or to a patient, remove the blood, test the blood, see what are the blood levels, and plot the required blood level curve. Instead of that, if you can see, these are certain physical models where this, if it is considered as a stomach, this various intestines, small intestine going into large intestines, rectum, and further excretion. These, each of the tubes are internally coated with the cell line of either the intestine or the stomach, whichever we are planning to do. The drug is inserted over here. There are various ports for withdrawing the solution, that is intestinal fluid or gastric fluid, and testing it. And ex vivo, that means not inside the human body, but outside the human body, these type of designs were initially created to arrive at the various algorithms and the software to develop into in silico approaches. So these were the designs which were used as PBPK model, and on the basis of this design, this is what has happened in silico. You have stomach, duodenum, jejunum, ileum, cecum, ascending colon, and final excretion. So you have the drug which is passing through these various media. There is an unreleased drug. So the tablet or the capsule is passing through. It gets dissolved. Some part of the drug is undissolved, passes into the next compartment. Once it dissolves, it goes into the bloodstream. It is absorbed. And after absorption, it goes into the portal circulation through the liver. And not just that, it goes into the other compartments like brain, adipose tissue, muscle, and skin. And this is how the whole compartment model is designed for an in silico approach or an in silico determination of drug release. And that is not just in fasted condition. You can also determine it in fed condition by just changing the pH. So initial studies, one has to determine what would be the pH of the stomach in, say, fasted condition, it is 1.3. In fed condition, it's 4.9. And accordingly, even the various pHs that we see existing in the intestine as the drug surgeons through that. So these approaches earlier, when the software was developed, one said, oh, it's a general software and you can't differentiate it between fasted and fed conditions and all. They developed further on it, and they created these additional designs where even fasted and fed conditions could be determined. And then this is just a social transit of a drug. Once it is put into the lumen, what happens? I'm not going to go into detail of all these things. So these phenomena are happening simultaneously in the body, and they are repeated in each and every compartment of the gastrointestinal tract, which I showed in the previous slide. And this is being captured in silico, and results are being provided through this particular software, which is Simulations Plus. This software was helpful to us, not just determining the kinetics of a drug, but also helped us in determining what would be the blood level of the final finished formulation, and whether that was adequate to provide the required effect to the patient. And and being in generic industry, we were also able to do virtual biostudies. Need not have patients or healthy human volunteers to carry out biostudies. We could do biostudies in silico virtually. And not just that, we could also do bioequivalence of the innovator product and our developed formulation, and prove to the regulatory agencies through this in silico approaches that we have achieved bioequivalence, no need to carry out human studies. Initially, there was a lot of resistance from the regulatory agencies to accept these data, but once they spoke to the company themselves and the company presented to them, they asked for some additional validations to be carried out, which all was done. And ultimately, the data was accepted, and we ourselves have got approval from the regulatory agencies through the data that was generated in silico. Imagine the amount of cost and time we could save by using this particular artificial intelligent simulation and modeling. Coming to process design, we normally concentrate on formulation design for quality by design, but we forget about the process. Sorry, even the process has to be done or designed based on QBD principles, not just process design. Now, even we are talking of analytical method development and design to be based on quality by design. So what happens in a process? You have input material, you have a machine, and you have output, which is capsules. I've taken an example of a capsule dosage form. Input material, of course, will be the blend, and you need to have CMAs defined for the blend. Machine parameters have to be set to get the output, which is capsules. These are the various parameters that one needs to study. Apart from that, you have to identify what are the critical ones. So we identify particle shape, flowability, moisture content as the critical parameters. Then you look at the various process parameters and look at what are the final process parameters that one needs to study. For the final quality attributes of the finished products, which we identified as the content uniformity is very important, the impurity profiling is very important, and of course the final dissolution. So after identifying these, one needs to chart out a plan whereby which how you will monitor how many experiments you will carry out through design of experiments. And if you see over here, the unit operation, that is the process parameters, needs to be controlled for your CPPs, which are a part of the complete product and formulation design. So we tend to study the straight line over here, but we do miss out in the additional circle that I have presented. This also needs to be done. And if you are having a good software by using quality by design and artificial intelligence, it's not a very, very difficult task to achieve. Coming to quality, on the quality front, we have various softwares which are available to us, and there are various instruments, equipments in the lab through LIMS, that is Laboratory Information Management Systems. One can link all the instruments, all the data is captured. LIMS calculates the complete data for that particular batch. LIMS will then pass on the information to another software, which is called as ERP or SAP. SAP will have in it the specification for the finished product. SAP will compare whether the data which is generated from LIMS is comparable to the specification, and SAP will decide about the release of product to the market. So this is very, very briefly on AI in labs, where labs can also become completely paperless, no human decisions required. The software will decide for you the fate of the batch. Coming to manufacturing, yes, excellent examples of automated warehousing systems where you have robotic driven automated storage and retrieval system where the material is stored in the racks. The computer and AI has a complete track of which material is placed where. And as and when you make a batch, you just feed the batch details into the computer. The computer will withdraw the materials from the warehouse and bring it out for your dispensing and then finally manufacture of the batch. This is just a brief view of a completely automated warehousing system, which we had also implemented and created in our facility. It's an excellent system. It also gives you feedback on how much material came in, at what stage is the material, whether it's under test, released, or approved, whether the material is now it's getting depleted and the reordering quantity has reached its level, so you need to order it again. All this information and feedback can be obtained through a complete software which is driving this. Now, this may or may not be considered as AI. Many a times people are challenged, this is not AI, this is just simple automation. Okay, fine, this may be simple automation, but it is then linked to other softwares which are helping to drive it. So I don't mind considering it as an artificial intelligence. Apart from this, on the manufacturing shop floor, currently PAT is the name of the game. PAT is process analytical techniques. It is helpful to show that the process is within the limits and the design space that you have created for it. How do you do that? You have various probes which are put into the various equipments, and these probes will capture the details and the data of what's happening within the equipment. Say, for example, you have a V blender. Now, whether the blend uniformity is achieved or not. In our days, we used to stop the blender, collect the sample, send it to the lab. Lab will test, lab will tell us no, uniformity is not achieved. We go back, we start reblending, blend for another 5-10 minutes, again stop it. That's all out. You have a probe within the blender itself, and the probe will tell you what is the end point of blending when the uniformity is achieved, period. Excellent process. And because we could go up to the extent of creating these PAT initiatives and having online or inline determination of the endpoint of the process, we have started going into the next step, which is continuous manufacturing. Now, before I go into continuous manufacturing, let me just talk to you about smart machines. We have currently smart machines on the shop floor, machines which once set and the parameters established in their software will continue to operate throughout the manufacturing of the batch within those particular set parameters. That doesn't mean that the machine parameters will not change. The moment the machine parameters start changing, the software will tell the machine, hey, look here, your average weight is shifting a bit. So the machine resets itself and brings the average weight back within the set limits. These are the smart machines, and these are the machines which are operable because at the back end, we have complete data analytics, we have complete softwares, we have complete algorithms, which helps these machines run and deliver a batch within the required specification. Now, imagine over here, earlier in our days, we used to sample from that particular machine every hour. Take the sample to the in-process testing lab, carry out weight variation, carry out DT, hardness, friability, and then allow the machine to continue. What a huge saving it is. What a huge saving. We are not, what do we do with the tablets that we collect? We have to discard them. And secondly, these machines do not require a lot of manpower. They run on their own. They are smart machines. And this machine gave a big advantage during the COVID times when we were running facilities on skeleton manpower. Just one person on the whole floor could manage continuously manufacturing a particular product. So as I said earlier, this is a brief slide on continuous manufacturing for using a roller compactor, where at each stage you are seeing the in-process parameters being checked and monitored through PAT initiatives, and we can achieve a complete single line process without stoppage as a continuous process. Only issue is even today we are having some problem with respect to dissolution. Dissolution, as you know, normally we go by weight gain, but that's not a parameter that one would like to achieve, and not a parameter that one would like to use as a final decision-making parameter for achieving the dissolution. We are looking at additional AI initiatives to try and find out what can be the best possible way for measuring the dissolution endpoint. What are the other advancements? I mentioned about 3D printing tablets. This is the first 3D printed formulation which is approved by FDA. There are smart gel printed organs which are currently now also being utilized, and some animal studies are already underway. And very soon we will be looking at personalized medicines through 3D printing, especially in case of cancer patients where they really need a personalized combination of products and a personalized strength for the type of cancer that they're suffering. So these are the various advances we see. The last part is the distribution, where I was talking about track and trace. Each individual strip is printed with a barcode. The strip goes into a secondary pack, which is a carton, which is again printed with a barcode. Many of these cartons will go into a shipper. The shipper has a barcode, and this barcode is linked to all the other barcodes of the products which are there in the shipper. The shipper goes onto a pellet, and the number of shippers and their barcodes are then linked to a barcode on the pellet. So this is how complete tracking happens from a single blister in the company up to the pellet, which is going out from the warehouse. And this pellet, when it reaches the warehouse for the supplier, and it reaches, and then these boxes, when they reach the retail pharmacist to the patient can trace back the product from his hands back into the company. And if at all there are any issues, the companies can do this. Very useful for investigating any market complaints. I know which batch this particular product belongs to. I know what's the expiry, and by scanning it, I know what ingredients have gone in and what could have caused the problem. It really helps in carrying out root cause analysis of the various issues which could be reported through pharmacovigilance. Now, this complete implementation deadline was given 31st March 2023. It was supposed to be 2020, but because of COVID situation, they went up to March 2023. But from what I read, it has further been extended to August. I don't think you can see this. It's further been extended to August of 2023. So this is what is AI. This is what is big data. We are talking of big data. This is big data at each stage of the product in the marketplace. This is the big data which is being generated and put into cloud. Coming to the last and final part, that is AI applications in regulatory, pharmacovigilance, product lifecycle, and ERP. Under regulatory part, there are a lot of softwares for the quality assurance and quality management systems, especially to deal with the corrective and preventive actions that a company is required to take. Once you have a particular problem in your lab or on the production shop floor, you have to report the issue. Once you report, say for example, you had a result which was out of spec in the lab, you have to carry out an investigation. You have to raise a CAPA. What is the corrective and preventive action that you are planning to take for that particular result which is out of spec? Once you have raised the CAPA, you have to carry out root cause analysis why it has happened, because unless until you know why it has happened, it's difficult to generate a CAPA. Now CAPAs could be running into a couple of years. Corrective action you can take immediately, but the preventive action you may have taken in a few months. But whether the preventive action is effective or not, whether the preventive action is sustainable or not, that needs to be determined over a period of time, for a number of months, for years, for a number of batches that you make. Unless and until that is done, your CAPA cannot be closed. So who will track it? Is it physically possible to do it? No, there are softwares available which will help with this. There are softwares available which will help you to manage your documentation and data. There are softwares available for data analytics, for continuous improvements, and even for training records. Training records, believe me, in regulated companies are very, very important. If a person is found to be working on shop floor who's not trained on that particular activity, all the activity carried out by that person is null and void. It may go to the extent of asking you to reject a batch because an untrained person has handled it. So training records are very, very important. And believe me, in the QA, quality assurance or quality compliance, very, very important on documentation and document management systems, because as and when we are being audited, the auditor expects you to have the data in your hand within half an hour of his asking. How do you manage it? When it was physical records, it was very, very difficult to get those data. But now being electronic and having electronic records on hand, it's difficult, but it's less time consuming. With a click of a button, you can go into the data that you are looking for, download it, and hand it over to the auditor who's requested for it. Coming to regulatory affairs, complete document submissions can be done through AI. You need to ask the R&D, the clinical department, the medical department, everyone to upload the data on the product which is being developed online into a particular software which the company has taken the license of. And once all that data is available to a RA person just needs to determine for which country the submission is required. Is it required for US or Australia or Europe? And based on that, it can collate the complete document for submission within a couple of days. Without this software, believe me, we in our company, we were taking months to collect the data. We had to run after each department to ask for the information, to ask for the data, determine that for this country, what all is required and from where do I get all this information, then collate it, review it, get QA'd, and then further complete submission can be put into the regulatory agency's pipeline. So currently with this AI-based software available, it's become really very easy. Even responses to the queries which come in from the agencies is very easy to give. You all must be aware, if you are working in regulatory space, that annual updates are a must. Each company has to provide annual updates on each and every product to the regulatory agencies on the complete happening, how many batches you have manufactured, whether there were rejects, whether there were any recalls, any market complaints. Complete update on each and every product, each and every batch has to be given to the regulatory agencies in the countries where you are operating. So this again is a huge exercise, a huge data. If you have AI in place, if you have document and data management systems in place, it becomes a bit easier. On pharmacovigilance, again, pharmacovigilance has now picked up even in India. You have people reporting from the marketplace, you have physicians reporting, you have companies reporting, you may have even finally the patients reporting on various issues that they have with the product. If you have a strong pharmacovigilance software, it's very easy to handle all these complaints and give feedback to the complainant in time. And not just that, have the data ready for submission in your annual updates. And not just that, as and when there is an audit and a request for any pharmacovigilance data to be provided, it becomes very easy to handle that particular data. So coming to a complete life cycle management of a particular product, from planning up to the final product into the market till its life is there in the market, you can manage it through complete life cycle management programs. We have the planning, we have the logistics and supply chain coming in, we have lab testing and release at the procurement stage, at the sampling stage, at the commercial manufacturing and final testing and release. And all these things can be done through an ERP system. We were using SAP. I think worldwide SAP is the one which is most popular and a very, very well validated system. Just one AI news which I had seen, it's excellent to read, that award-winning mobile app was developed in Tamil Nadu in our own country, and it's artificial intelligence based. It can identify cataract spots with 91% accuracy. What a this thing, happiness to read such a thing happening in our own country, that you are even able to diagnose using artificial intelligence. So all this was very, very positive aspects of artificial intelligence. Just going into a bit of the negative aspects of artificial intelligence, and you all are aware of the viruses which are coming in. You are all aware of the various cybersecurity concerns which have happened during the COVID times. On a regular basis, many pharma companies came under cyber attack. In India, we had Dr. Reddy's who was under cyber attack in October of 2020. And all this also happens through artificial intelligence. And this can be once again countered via artificial intelligence itself, having a very, very robust cybersecurity cell within the organization who is able to counter it and track it down. So there are two very big challenges within any organizations today. One is implementation of artificial intelligence. And believe me, once artificial intelligence gets so prominent, we land up with something what I always talk of as natural stupidity. Today, we are talking of GChat, and people are saying, why are you even making slides? Tell GChat. GChat will tell you everything what needs to be put onto the slide. Now what happens to me? I'll become naturally stupid. I will depend totally on AI and all the AI options available. I'll not use my brain at all. And this is something which is really, really very challenging. And in order to implement this in a balanced level, one needs to create such a mindset within the organization. And what I would like to say is that although we are talking so much of artificial intelligence, nothing can replace the human touch, because it's always the human brain and the human mind which is behind artificial intelligence. Those people will become enriched, whereas people who are just using it on face value will become stupid. So we need to go a bit beyond what's there in front of us and try to learn how to adapt to it and work in harmony in order to be successful. So artificial intelligence and machine learnings have transformed the way in which we are doing a lot of business, but the implementation is very, very challenging. And the main challenge, friends, in our pharma industry is regulatory compliance. Regulatory compliance, mainly with respect to validation of all these softwares which are AI-based or machine learning or robotics or cobots, whatever you want to implement. Pharma industry requires validation. And that is a very, very big challenge. Why do we think that a simple aspect of track and trace, we in our company, we took 3 years to implement because of the validation challenges. At each stage, one has to validate it, not just to determine and prove that the software is doing what it is required to do, but also to prove that the software cannot do what it should not be doing. So you don't just positively validate it, but you have to do even a backward validation by negatively challenging the software and checking whether the software stops you in your tracks and does not allow you to proceed further. So this is a bit from my end. I think I would like to close now. So wishing everyone all the very best. Once again, thank you for giving me this opportunity to talk to you. You have any questions, this is my contact details. And all of us together, teachers and academicians and academics and industry, we all need to get together and keep the India flag flying high. So thank you very much.
Okay, thank you, ma'am. The bottom line is important. Nothing is replaceable than human brain. Okay.
Yes.
With this tagline, I would like, ma'am, some questions are there in chat box. I would like to put that questions. The very first one is coming from the audience side is that how do we get access to the software like DDD and GastroPlus?
Yeah, you have to buy licenses for those softwares. And even if you don't want to buy the softwares as academicians, I think you can request these softwares are available and distributed in India by Electrolab. You all are aware of Electrolab, right?
Yes.
Yeah, so Electrolab is the main distributor for this software and providing the required licenses. So you can get in touch with them. They will help you out.
Thank you, ma'am. Very next question is, do we get odd result in spite of implementing QBD? And what is the use of design space if the kappa still features? I don't
do we get odd result?
Odd result in spite of implementing QBD?
Yes, you can get odd results. We call them outliers. Okay, so if
outliers, okay.
yeah, those odd results, what you're talking, technical term is outliers. So if you have an outlier and if you can justify that it is an outlier and you would not like to consider it in your quality by design data which is taken for statistical processing, it's okay, you can go ahead and do it. But whatever decision you take, I hope everyone understands risk-based analysis. Those decisions have to be risk-based. You have to carry out complete impact analysis of that particular decision, how it's going to impact your final results. And once you feel that the risk is low, you can remove that outlier from your particular QBD. But if that outlier is not an outlier and is a true result, once again, carry out a complete risk-based analysis and see what is going to be the impact of including that result into your QBD. So QBD data is huge. So many a times, like maybe a single outlier, even if that data is not with what you are expecting, it may or may not impact the complete data on a statistical analysis basis. I hope I could explain.
Yes, ma'am, sure.
Okay. What is the use of designs? This is the next part of the question. What is the use of design space?
Yes.
If kappa still features, can you explain, given an example? I mean, these are two different things. You are developing a design space as and when you are doing product design or a process design. At that stage, you do not expect a kappa is coming in. If there is a kappa, then you cannot proceed further. Like say, for example, you wanted an example. You are expecting a disintegration, which has to be not less than 10 minutes. And your disintegration test is not coming to 10 minutes. You have an outlier, or you are going out of spec and your DT is coming to 15 minutes. So you can't even move to your design space because you're not achieving what is required to be achieved. Right? As I explained previously, we would develop a product, and if continuously I'm getting DT 15 minutes, my spec would be 15 minutes. But for quality by design, your 15-minute spec is not acceptable. Through QBD principle, you have to design your product to get a 10-minute spec. So your kappa is still there. You have to take a corrective action to develop a design space with a specification of 10 minutes. So you have to go back, correct yourself, correct the design space, and then only will you be able to close the kappa. But to close the kappa, you have to find out what is the root cause of you not able to achieve a 10-minute disintegration time. So you have to carry out complete root cause analysis, identify the root cause, correct the root cause, then go back and work on your QBD program. So it's the other way around. Okay.
Okay, ma'am. Thank you. I hope most of the ideas have been clear, but this is actually totally new field, and there is lot queries in our mind, ma'am. And I think in most of the sector that you have explained, from drug discovery to the manufacturing or regulatory affairs. So every step need one deep lectures like that, today's one. So it is a very broad scope we are having in this new era, and with hope that you will be available for us whenever we need for the next session also, ma'am. Thank you once
Sure.
again for your
Yeah,
wonderful
most
session.
welcome. Actually,
Yes,
I mean, this is almost a full 1 day talk, you know.
Yes.
yes, yes.
Yes,
And I
it
was
is.
asking Dr. Joshi, you given me only 40 minutes. Can I extend a bit? Because if I have to cover everything, then
Yeah.
Yes.
this 40 minutes is not possible. So thank you very much for this additional time. I think I have gone beyond the time that was allocated to
No,
me.
ma'am, but like it's absolutely fine. Actually, we could also get an overview today, and for details, of course, we will get connected again, because
Yes, ma'am,
yes, it's very
definitely.
deep, and we could get an overview though, of course.
Yes,
Excellent
yes,
session,
yes,
ma'am. Excellent.
Right,
yes,
right.
Complete different thought process in our minds also, like.
Right. So just to give you an example, you know, our development time, which we used to be from 5 to 7 years, we could
Okay.
bring it down to 3 years. 3
Oh,
years, yeah. And to add to that, once again, reduction in time for approval from regulatory agencies. So
Yes.
it's so fast. So although people say the cost of these softwares is very, very high, you can reduce it by reducing your development time. And for academia, you can approach these people directly. Like you can approach Simulations Plus and say that we want you to present your software to our
Software.
academic session. They will do it. You can invite them directly.
Okay.
we will
Right?
do.
Definitely,
Yeah,
ma'am.
yeah, yeah.
Yes, yes. But what happens, ma'am? Okay, these engineering people, they are supposed to be higher of this AI and instrumentation and all. So we need to be do more in this field, and the people like you are only going to help us.
No, they will. If you
what.
tell them this is our background, they will come to your level and talk to you.
Yes, yes, ma'am.
They will definitely, yeah. Yeah. So they will talk to you on the application side. They won't talk about the engineering part and all.
Engineering part. Yes,
Yeah,
yes,
yeah.
that's right.
Hmm?
Okay. Thank you very much, ma'am. This
Most
is very
welcome.
valuable knowledge that we get from you.
Thank
And
you.
we are very thankful to you for your valuable time and presence, ma'am.
Thank you.
Thank you very much once
My
again.
pleasure. Thank you.
Yes. Now I think Dr. Anagha, ma'am, we should go for second session.
Yes, ma'am. Immediately.
Yes, definitely.
Yes,
Okay,
yes,
okay.
yes. Please
Okay.
proceed.
So with this huge knowledge, we are switching over to the second session. I would like to introduce our second speaker of the second session of today's webinar series. She's Dr. Aarti Prabhu is a drug discovery scientist and medicinal chemist. She received her PhD in pharmaceutical chemistry from Mumbai University and has over 20 years of computational chemistry and medicinal chemistry experience in drug discovery area. Her contribution has earned her several publications and one patent. She is currently working as an associate professor at SVKM's Dr. Banuben Nanavati College of Pharmacy. Her research interests involve developing targeted drug therapies and technology. She is a recipient of around 40 lakhs research grant from universities, AICT, DST, and RGSTC. We are very happy to see you, ma'am, for the next session. Over to Dr. Artipal.
Thank you, madam. It is indeed a pleasure and honor for me to be talking in this forum.
Yes,
let me just share my screen.
Yes, ma'am, you can share. You are a co-host now.
Yeah, sure. Okay, so good morning.
Yes, ma'am, it is visible.
Yeah, thank you. The topic which I have chosen today is 3D therapeutic thinking, focusing primarily on computational tools which are used to aid in the design of therapeutic drugs. And I would really like to thank our previous speaker, Dr. Parizad, for already setting the stage for me, while she gave a fascinating overview of the entire process, AI from molecule to market. I will be focusing on some widely used computational techniques which are used for the first part of what she described, for the design of active molecules, for new drug discovery or molecular discovery, as what Madam has described. And hopefully I will be able to give you a flavor of how, when we look at molecules in 3 dimensions, let me see if I can take my laser. When we look at molecules in the 3 dimensions, it makes them more real, more closer to the biological context. And thus, when we try to design drugs using the 3-dimensional structure of molecules, it makes our efforts more focused on success. So this will form the agenda of my talk today. So I'm zooming in on how we can use computational tools, or if I have to use the terminology of the previous speaker, AI, for discovery of new drugs. So these are some of the widely used techniques which are used for new drug discovery. I will elaborate with examples, some techniques, some tools, or some methods which come under structure-based drug design, or as it is called as SVDD. Under this, I'll be discussing how we can use the binding site for guiding or driving the design of small molecules. I will briefly touch upon the principles of homology modeling and docking. So these are widely used or widely known techniques which come under docking, or which come under modeling. So whenever somebody says CAD, the first thing which comes to mind is docking, and the second thing which comes to mind is homology modeling. So both of these techniques come under structure-based drug design. So I will try to give some examples, describe the basic principles, and then give some examples how we can use these tools to accelerate drug discovery. I'll also be talking about some ligand-based drug design tools, or LBDD techniques, primarily just touching upon similarity searches and how we can use that to do virtual screening. And finally, at the end, if time permits, I would like to touch upon something which is quite interesting to me at the moment, how we can use binding site waters or active site waters to design to improve the potency of small molecules. So this is what I have planned for today. So starting with structure-based drug design. When we say structure-based drug design, we are actually referring to the structure of the target. Okay, so although the small molecule also has a structure, which is extremely important, by convention, when we say structure-based drug design, we are referring to the structure of the protein target, okay, the macromolecule, the receptor or the enzyme, which we want to target. And when we use the structure of the protein to guide the design of the small molecule, guide the design of molecules with optimal fit, that comes under structure-based drug design. So it's very much obvious that if we have to use this approach, the SVDD approach, what is essential, what is required is the 3-dimensional structure of the protein. Can we get this? Can any person get this? Yes. even if you do not have software. So what Parizad madam has correctly pointed out, that we need to license out the software, and it may not come cheap. We may have to take a demo license, as she described for Gastro Plus or Simulations Plus. We have to take the license for it, we have to pay for it. But assuming we have nothing, we have no license, nothing, still we can bring in some amount of 3D insights into our designing process, because the protein structures, the 3-dimensional structures of proteins, are readily available to all of us. It's a public resource, an open resource. In fact, most of the proteins, not all, but most of the proteins can be crystallized, okay, and their structures, the 3-dimensional structures, can be elucidated by some analytical techniques. Commonly used technique is X-ray crystallography, X-ray diffraction. So using this technique, most of the protein structures have been solved. The coordinates of the protein are available. If not X-ray crystallography, some of the proteins cannot be crystallized unless they're in the biological setting. So for example, membrane proteins, they will lose their structures if the membrane is not there. But there's another technique, if the proteins are smaller, if the scale is smaller, the solution structure can also be elucidated using techniques like NMR. So there are a couple of techniques which are available, which are used for determining the 3D structure of biological proteins. And all of these are deposited in a public database, the Protein Data Bank. I'm sure most of you are aware of this resource. So if you visit the Protein Data Bank, then you will see that there are millions of proteins as well as nucleic acid structures which have been solved and deposited, available for download, free. Okay, so this is the go-to resource for downloading the PDB structure of the target protein, which we can use for SVDD. How do we look at it? So you downloaded the structure, the .pdb structure. There are some freely available visualizers. So they will not help you to run any simulations or job, but something like PyMol or Discovery Studio Visualizer can be freely installed on our machines, and we can open the .pdb file and we can move it with our hands. We can look at the active site, we can study the active site, we can get a feel of the properties of the active site. Is it hydrophobic? Does it have hydrophobic amino acids with some potential for hydrogen bonding? Is it a shallow pocket? Is it a deep pocket? Are there different conformers of the inactive and the active protein? So we basically get a feel of the ground. What is it that we want to target? And once we have that idea, then we can go towards our designing or synthesis or screening efforts with this insight in mind. So this is basically something which I encourage all the researchers who are into medicinal chemistry to do. Even if you do not have licenses, download the PDB structure and have a look at the active site, read up its mechanisms, and then you get a feel of where you want to focus your lead identification efforts to go. These are some of the well-known examples, success stories which have come out of SVDD. So there are many. I have just put 5 over here. Dorzolamide, for one of the first success stories of SVDD, carbonic anhydrase inhibitor used for reducing intraocular pressure. Sakinavir, HIV protease inhibitor. Zanamivir, neuraminidase inhibitor used for influenza treatment. Imatinib, a tyrosine kinase inhibitor used for various cancers, especially CML. And Odanacartib, I have put this here because I have a case study which I'll be describing of Odanacartib. It's an anti-osteoporotic drug. Okay, so you can see these are some of the molecules which have been developed by using the protein structure as a guiding point, the binding cavity, to drive the design of these molecules. So my entire talk is interspersed with examples, because basically I want to exemplify the tools. Okay, there is a lot of theory behind it. The computer always has an algorithm which has been developed and which is constantly updated by the application scientists of those software companies. But as end users, we want to know what is possible to start with. Okay, so this is the story of Odanacartib. It is a cathepsin K inhibitor, CAT-K inhibitor developed for treatment of osteoporosis. So you can see the structure. It has a biphenyl and sulfone SO2 Me group, and it's quite a long structure. Cathepsin K, just to give one line, the context of why CAT-K inhibitor for osteoporosis. Cathepsin K is the predominant bone degrading protease, which is responsible for worsening osteoporosis. So if we can inhibit cathepsin K, that would be an ideal target for anti-osteoporotic drug development. As I said, we are looking at SPDD. So the first primary requirement is the crystal structure available, is the structure. Let me not restrict myself by saying crystal structure. Is the three-dimensional structure of CAT-K available? Yes, so it was available. The binding cavities, there were multiple binding sites. The catalytic site, it's a protease, so catalytic site. Allosteric sites were all thoroughly studied, well mapped out. So that allows us to use the SVDD technique for optimizing or developing drugs, optimizing or developing cathepsin K inhibitors. So,
screen कुठे आहे? Line गेली ना.
Hello.
Net change झालं.
Okay.
Manisha madam, please mute yourself.
Okay, sorry.
Thank you. So Odanacartib was developed by Merck, and I want to put the news report here to start with. Odanacartib had such robust efficacy that there was a recommendation from the clinical advisory panel while it was in the clinical phases to stop phase 3 studies earlier, because the molecule was found to work much better than expected. It was a highly potent, highly effective drug with a good safety profile. Okay, so that was so that's why we can say Odanacartib is a success story. However, point to note, Odanacartib was not the first clinical candidate. The first clinical candidate was L006235, which also had excellent efficiency. Its cathepsin K IC50 was 0.2 nanomoles, sub-nanomolar potency. But when it was taken through the clinical studies, it was found that it had two major drawbacks. It was toxic, and the toxicity was attributed to this basic nitrogen. And there were two amides in the structure. The central amide was hydrolyzing very fast, and because of which the short life of this anti-osteoporotic drug was very, very short. So because of these two drawbacks, the molecule had to be optimized. Okay, so at this point, it is already in clinical, it's a clinical candidate, meaning the potency has been optimized both in vitro and probably in animals. But at this point, when we realize that there are some drawbacks which would kill the drug, there is need for optimization. So the first clinical candidate was having high potency, but toxicity and a very, very short duration of action. So when they probed this mechanism, it was realized that the molecule, the basicity of the molecule, the basic nitrogen was forcing it to accumulate in acidic subcellular compartments, not only in the bone, but also in other acidic compartments where it was inhibiting other functionally required cathepsins. So cathepsin L, cathepsin F, cathepsin S. It was having a number of off-target hits. And because of that, that was resulting in a lot of serious adverse events such as cardiomyopathy, impaired neovascularization, and even the immune response was getting affected. So the thing which was bringing down this molecule was the basicity of the molecule. Secondly, the amide bond the central amide bond was liable to rapid metabolism. So it is obvious that both of these structural features had to go, but because the molecule was already optimized for potency, one had to be sure that these features were not contributing to that 0.2 nanomolar potency. Because if you just replace this with the hope of removing the toxicity and improving the duration of action, it's highly likely that you may lose the potency and you would have to once again take it back to the drawing board. Okay, so for this now, SVDD comes into picture. So since the crystal structure was available, the molecule could be docked into the structure, into the binding cavity, and it could be assessed what is the contribution of the basic nitrogen. So this is the piperazine molecule, which has 2 nitrogens. So you can see here, this is the piperazine molecule. This is the nitrogen which is outside. This was found to be solvent exposed, but the inner nitrogen was actually making a binding interaction with the aspartic acid. So if we completely replace this piperazine, this interaction was likely to be impacted and the binding affinity would have come down. Secondly, the central amide. So you can see here CONH. So this NH was actually making another crucial binding interaction, a hydrogen bond with the backbone of a glycine. So if you replace the amide, it is likely that this hydrogen bond will also be impacted and the molecule would lose its affinity. So it is obvious that the amide had to go. So what if the double bond O was removed? If the double bond O was removed, this molecule would become a base, once again giving accumulation in acidic compartments and inhibition of other cathepsins. So all of these implications had to be taken into consideration, and a strategy was made for lead optimization. A structure-based strategy was made for lead optimization. So the main thing was keep the NH because it is forming the hydrogen bond, get rid of the C double bond O because it is not really making any interaction. But if you remove the C double bond O, because the basicity would increase, put an electron withdrawing group. So using the principles of chemistry, if we put an electron withdrawing group on this carbon, it would still be an amine, but the basicity will be reduced. Even if it's weakly basic, it would still form the hydrogen bond. So that was the strategy which was used for dealing with the central part, the central amide. But there was one more aspect which had to be considered. Adjacent to the center. So if you replace this C double bond O with a group which is electron withdrawing, it was likely that it would clash with the adjacent cyclohexyl group. So that means this rigid lipophilic cyclohexyl group also had to be replaced with a lipophilic flexible, probably an aliphatic. So using these insights, the CONH, the NH was retained and the C double bond O was replaced with CCF3. So this is being an electron withdrawing group, would decrease the basicity of this nitrogen. So this is the overall strategy which was made. The piperazine was replaced with SO2Me. You can see here that the nitrogen which was making the crucial hydrogen bond, that hydrogen bond could still be got because the SO2, the double bond O, is polar enough to make the hydrogen bond. So now this SO2Me is not at all basic, but it has the capability of forming the hydrogen bond. The thiazole was replaced with phenyl. The NH was retained, but the C double bond O was replaced with CCF3. The cyclohexyl group was chopped, and we have another lipophilic flexible group in its place. And this particular group was sort of frozen by putting a cyclopropyl group here. That was another strategy which was used to sort of force the bioactive conformation. So there are multiple changes from this first candidate to odanacartib. If these changes were done without using the structure of the protein, the binding site, as a guiding point, at any point, the affinity would have been lost. But because the docking protocol was in place, the first compound having 0.2 nanomolar IC50, and the fourth step down, the molecule which was odanacartib, you can see the IC50 was retained in spite of completely changing the molecule. So this is, and moreover, the toxicity was gone. It was no longer basic, so it was not accumulating in any of the acidic compartments, subcompartments. Secondly, the half-life. The half-life was so good that this molecule could now be given once a week. So once a week dosing is permitted in odanacartib, and that is quite a boon for a person who is suffering from osteoporosis, a chronic disease, and therefore if you have something which is having a long duration of action, that completely changes or improves the patient compliance. Okay, so this is a success story of structure-based drug design, where the structure was available, docking protocol was set up, and changes were made. The initial lead was optimized. So this is an example, a success story of lead optimization to have retention of the initial potency, but the toxicity was mitigated, the half-life was still in. So it was drastically changed to something which now gives you a better patient compliance. Now, what if the structure is not available in the PDB? Okay, so supposing for some reason the structure cannot be, the protein cannot be crystallized, NMR is not possible, probably because if the protein is very large, then NMR gives such a cluttered, chaotic spectrum that we cannot elucidate a structure. So if these experimental pitfalls come and we don't have the experimentally elucidated structure of the protein, then what does one do? So one can then turn to homology modeling. This is another computational technique widely used. Here we can build the structure, the theoretical structure of the protein can be built by using principles of bioinformatics. All you need is the primary sequence. So all proteins have a primary amino acid sequence. If you have that sequence, then it is possible to go through the process of homology modeling and build a theoretical model for your docking or other binding site analysis, etc. Approaches. So here, in very brief, what is done? The first thing that has to be done is you need to identify a template molecule, a template protein whose 3D structure is known and with which our primary sequence aligns well. Okay, so at least 30 to 40% of sequence identity or sequence similarity or sequence homology of the query sequence in which you are interested in should be there between the query and the template. Secondly, the template should have the 3D structure elucidated value. So then in those regions where the sequence of our query molecule and the template are aligning, in those regions, one can directly copy the structure of the protein onto our query sequence. And in regions where the structure, where the sequence is not aligning, okay, there you need to extrapolate the structure by various techniques, one of which could be loop modeling if it is part of the loop. Okay, so there are various, again, theoretical algorithms whereby we can extrapolate the missing chains. Once the backbone of the protein is built up, the side chains. Okay, so all of the amino acids will be side chains to the primary backbone. So the side chains then are sort of adjusted with one another because so as to prevent clashes with the neighboring amino acids. Once that is done, the model is then the entire model. So now we have built the structures, we have extrapolated part of the structures, we have explored the rotameric library of each amino acid so that they are not clashing with their neighbors. And this raw model is then optimized by various, again, theoretical tools, one of which is molecular dynamic simulations. The whole molecule can be perturbed either in vacuum or in water, or if it's a membrane protein in the membrane environment, so that it becomes energetically stable. So this model then has to be validated. There are again a number of parameters for validation. Once it is validated, if it satisfies the criteria validation, then we can nominate it as a validated homology model and take it for our docking studies or other structure-based approaches. If we do not find validation, it needs to go back into the iterative cycle so that we can come up with a more geometrically feasible and functionally viable structure. So now, having shown you all two preliminary approaches, either we go to the PDB, download the PDB, or if it is not available, if our target of choice is, if the target of our interest is not available, the structure is not available, we can build the homology model. So having the protein in place, having studied the binding site, now we can look at other techniques, other advanced techniques, one of which is docking. This is, as I said, docking is synonymous with modeling. So whenever somebody says CAD, the first thing which they say is, "Acha, docking karna hai." So this is one of the most popular, or the face of modeling, if I can say so. So again, it's a theoretical algorithm. The docking program, the program, the script of docking does not know the biological context. It does not know that the endogenous or the native ligand binds in so and so position, induces the conformational change, and the signal transduction happens. It doesn't know that. It is just an algorithm of brute force. Okay. So what is done? If this is the small molecule, the small organic molecule, which you want to fit in this active site. Okay, and you want to find out which is the most likely position of this molecule here, and that we nominate as the bioactive pose. So what is done? First and foremost, a small molecule is not a rigid molecule. Every molecule has flexibility because it has a number of rotatable bonds. So each bond has 360-degree freedom to rotate. So if you have like you have two bonds can rotate through 360 degrees. So you can imagine a regular small organic molecule having 8 to 9 rotatable bonds will have exhaustive or huge conformational flexibility. So you and me, we cannot like draw these or sketch these conformers, but the computer can for sure. So the conformer library of this molecule is first generated. Each rotatable bond is rotated through 360 degrees, and a set of conformers is generated. Then each of these conformers is positioned in every possible orientation in this binding site. Okay, so we have to define the binding site, and in that region, the molecule, the query molecule, small organic molecule, every conformer of this molecule is placed in every possible orientation in this active site. And for each of the positions of every orientation of every conformation, the interaction energy between the protein and the small molecule is computed by a formula. Okay, the energy equation, which is known as a scoring function, that the energy is calculated or computed. So this is known as a search plus score algorithm. The binding site is first searched, and every possible orientation is scored by the scoring function. Then all that remains to be done is rank order the scores, and that particular orientation of that particular conformation which has the least energy, the most stable interaction energy, the most negative energy of interaction, that is nominated as the bioactive pose. So in very short, we can call docking as a search and score algorithm to nominate or find the most likely bioactive pose. So it goes without saying that if you use softwares which are not doing an exhaustive search, then you are likely to miss the bioactive pose. So many times we say ki the docking is not giving me correct results. Okay, it's not predicting the correct pose, not even of the native ligand. So one of the problems could be that the search is not exhaustive enough. Secondly, the scoring function may not be complete. So if you use a scoring function of some softwares which have not updated, okay, they are not taking every part of the energy function. They have only taken, say, for example, hydrogen bonding and hydrophobic contacts, but they have not taken into account desolvation penalty, okay, or internal strain, or hydrophobic entrapping. So there are various factors which contribute to the energy of interaction. So if you're working with a free software where the scoring function is not accurate, not validated, not tested, then you are again likely to come up with some wrong results. And then you may feel, oh, docking doesn't work. Okay, so that is the thing about docking. It is basically an algorithm which is using a lot of maths at the back end. So we need to have a software which has worked on that mathematics. Just one example to exemplify a success story of how we can use docking to identify a lead molecule. This is the homology model of the alpha adrenergic receptor. So I have purposely taken an old paper, 2005 JMedChem paper, because at this point, the crystal structure of the alpha adrenergic receptor was not solved. The homology model was available. In fact, the homology model was built by the company who was doing this project. Aventis had built this homology model and published it in JMedChem. So this is another approach of how we can use docking. Most of us who are in the academic setting, we design molecules. We look at the native ligand. We say, oh, it's a benzimidazole with this and so side chain, whatever, biphenyl, all of that. We try to make a similar molecule and we'll just dock one ligand. Probably we'll make an SAR. Around it, and we will go ahead. So this is another way that we can use the docking program. In fact, it is used in most of the pharma industries who are using, who have a molecular modeling department. Aventis had a large compound repository, and that is again a very usual practice. Whenever any research organization or research lab has worked on a lot of projects, you are sort of creating chemical matter, and you tend to put that in a database or a library. You never like leave it. You don't reject it. You always hold on to it, because when a new project comes, you would like to explore. Do I already have chemical matter for my new project? Okay, so Aventis compound repository had a large number of molecules. I could not find the real number, so I have not put here. But they wanted to see, do they have any alpha adrenergic modulators in their compound repository? First thing that they did, they filtered out a few molecules. They realized when they studied the binding pocket that the pocket is quite small. It's quite constrained. So there was no point looking at molecules which were large or highly flexible. So they filtered out the molecules which were large. They only took the molecules which had molecular weight less than 600 and rotatable bonds less than 9. And they applied a few pharmacophoric filters as well. And promptly they brought down the number to be screened by docking to around 23,000 molecules. Okay, then the 23,000 molecules which fit these criteria, all of them they docked using the gold software available from CCDC. Now, out of the 23,000 molecules, they found that 300 molecules had good scores. So they had potentially a good potential for binding to alpha adrenergic receptor. However, when they analyzed these 300 molecules, they realized that many of them had the same scaffold. So they did a scaffold-based clustering, and they realized that the 300 molecules actually belong to 80 diverse chemical scaffolds. So they clustered them, they grouped them into different chemotypes, and they took one best representative from each of these chemotypes, and only 80 molecules. So these 80 molecules, they tested experimentally in the radioligand displacement assay. So you can see from the large repository, using this workflow, they identified a smaller set of 80 molecules to be experimentally tested. And they found 38 molecules with promise. 38 molecules showed more than 50% inhibition at 10 micromoles. For these 38 molecules, now not for the 80 molecules, but the 38 molecules, they determined the binding affinity, Ki was determined, and they found that around 13 molecules had 10 micromolar affinity, 14 had around 1 micromolar, but 8 plus 2, around 10 molecules had nanomolar potency. Okay, and one molecule was actually a 1.4 nanomolar hit. So you can see the power of an algorithm like docking. If you use it wisely in your own compound database, your own intellectual property, okay, your own chemical matter, you can do a docking on a smaller set, experimental assay, the functional assay on a smaller set of molecules, and you may come up with a molecule which has excellent potency. So this is an example, a success for rational drug design. Okay. Now it's possible that their library was sort of biased towards the alpha adrenergic receptor. Probably they were working on GPCRs because the alpha adrenergic receptor is a GPCR, and therefore it's possible that 300 of the molecules, or let me come down to the experiment part, 38 molecules had good binding affinity. This is not always the case. So for every one success story which gets published, there are many which fail. But nevertheless, it is worth trying because one doesn't have to put in a lot of effort. Okay, but the point I want to make here is if you are doing virtual screening, okay, now virtual screening is analogous to high throughput screening. HTS is done in the lab, whereas virtual screening is done on the computer. If you do virtual screening based on docking, as was done over here, it is not high throughput. It is still, we can say, medium throughput. Why? Because if you have a library of even 23,000 molecules like they had, you have to do the conformer generation for each of these molecules. You have to prepare the protein. You have to do the docking, the actual search and score algorithm for each of these molecules. And then you will come up with those, whatever 300 hits. So the throughput for a docking based algorithm, if you're doing virtual screening, is medium. And that brings me to my ligand based drug design approach. So I'm just not going to really go deep into the ligand based approach, but just want to compare the virtual screening of a structure based approach with a ligand based approach and show you the tremendous saving in time. So what is ligand based drug design approach? This approach is used when the structure of the protein is not available. Okay, so for whatever reasons, if the structure is not available, if you cannot even build a homology model, so there is just no way that you know what the ground is like. You have no way of studying the binding site. But if you have some active molecules, okay, which have been tested, and you know that these molecules are active, you know the conformer of the active molecules, small organic molecules, then we go for a ligand based approach. Okay, there are several approaches like similarity searches or bio-isosphere searches, pharmacophore mapping. There are a number of approaches which are all bundled into this approach known as ligand based drug design. So I'm not going to really explain much of it today, but I want to make you aware of it. So the principle behind it is, if you have an active molecule, you know its conformation is such that it is active, even if you don't know how it binds. If you're able to identify another molecule which has a similar shape, okay, then it is possible that it would bind to that active site in the same way, and hence it would have the similar function. So this is known as the neighborhood effect. A molecule which has similarity with the active molecule is likely to have similar binding and hence similar function. Now the similarity could be on any parameter. Okay, the similarity could be on shape, the similarity could be the electrostatics, the similarity could be pharmacophore, anything. So if two molecules have some sort of similarity metric between them, then it is likely that they may have similar properties, similar binding, similar function. And if you have to assess the similarity, like when we did structure based, you had to do the docking to the protein. But here you are only calculating the similarity between two molecules. So it is very fast. All you need to do is try to align your query molecule to the active molecule. And then if it is similar, that's the end of the story. You say, oh, yeah, these are similar. It's likely that they will have similar function. And this is also one of the effective methods of doing scaffold hopping or lead hopping. From an active molecule, if you're able to identify another molecule which is similar to it in various attributes, you can move from one scaffold to the other. Other advantage of using a ligand-based approach is you can combine 2 or 3 different models. When we did docking, it was only one. How does this small molecule bind to this one target? But when we do a ligand-based approach, you can combine the shape model, the shape similarity model, with the pharmacophore similarity model. Now, the similarity value will go from 0 to 1. So you just get like either it is 0.5 similar or 1 similar. So if you're using like 2 parameters, the shape plus the pharmacophore, your score would go from anywhere from 0 to 2. Okay, so in this way, you can identify molecules which have the same shape and the same pharmacophore, hence more likely to bind to the target of the active molecule. So I'll end the ligand-based approach by showing how it can be used in virtual screening or lead hopping, as it is called. This is an example of a natural molecule. So this stachyflin was a natural compound from Stachybotrys. It had potent anti-influenza activity. So the researchers here wanted to identify different molecules or different scaffolds which were more amenable for synthesis, or from which series could be synthesized. Now, this particular natural molecule, it would be very difficult to make a series of this to understand the SCR. But so they wanted to identify other molecules which had similarity and which were amenable for synthesis. So they started with a database of 3 million compounds. They did a sequential sort of a filtering. So they did not use a combined shape plus electrostatics model. They first applied the shape model, and they realized that in their large library of 3 million compounds, it's only 5,000 molecules which had the same shape like this. This is a rigid molecule, so using shape does make sense. And of those 5,000 odd molecules, 8 molecules had the same electrostatics. So just 8 molecules were tested in the cell-based antiviral assay, because it's an anti-influenza project, and they found 2 molecules, 2 diverse molecules, 2 different series, 2 different scaffolds, which showed potential for optimization, more than 50% inhibition at less than 5 micromoles. So now they have 2 series, they can develop 2 series, one main, one backup, and they can continue their search for anti-influenza compounds. So finally, I hope I can stretch my talk just by another 5 minutes, madam. Okay, let me continue.
Sure.
Yes, ma'am, sure.
Okay, so this is dealing with active site waters. What it means. So up till now, I have shown you the protein, the bound ligand, and we tend to assume when we say a protein ligand complex, we are completely, most of the times, oblivious to the fact that there's a third component here. There are some waters which are trapped or which are part of this complex, which also contribute to the binding affinity, because the ligand and or the protein is forming some hydrogen bond to the water. Okay, so the third partner in the complex besides the protein and the ligand is the water. And most of us, even me, many of the times we completely omit the water. In fact, if you use something like Autodock for docking, Autodock cannot handle waters. It will ask you to delete all the waters. Okay, so we are looking at only 2/3 the picture. But those of us who can look at the water network, can actually use it to get a boost in binding affinity. Okay, so this is just a three or four slides on binding site waters. So before the complex is formed, the protein, the apoprotein, is solvated. The ligand before the complex is also solvated. So when the protein and the ligand have to come together, they first have to undergo this process of desolvation. So there's a desolvation penalty which has to be paid by both the protein and the ligand, after which they can come together, and the protein ligand complex once again gets solvated because it's in a liquid medium, in the extracellular or the cytoplasm or wherever. Once again it gets solvated. Now if the energy, energy of the solvated protein ligand is at least equal to or more than the energy of the separated, isolated protein plus ligand. Okay, if the complex plus the water energy is more negative than the individual, then we can say that this complex is stable. Water aids binding of the ligand. But if for some reason, after the complex is formed and solvated, if the energy, the magnitude of the energy is less than the sum of the solvated protein and ligand, then the complex will be destabilized. And here we can say that water hinders the binding of, water hinders the binding, the complex will not form. So even if the protein, even if the ligand is forming a stable set of interactions with the protein, if water is decreasing the energy component, then the complex will not be stable as a whole. And then we say water will hinder the binding. So this is just an example of an EGFR receptor. So this, what I have shown here, is the receptor. You can see these faint gray lines. This is the amino acids in the EGFR receptor. So when the EGFR receptor without the bound ligand was crystallized, you can see that there are some waters in this. And the color coding in my slide is pink is unfavorable, golden or yellow is favorable. So you can see here that when EGFR was crystallized, there were some waters which got trapped inside the active pocket. And these waters were not happy. Probably the active site is more hydrophobic, and the water molecules cannot make any hydrogen bonds in this hydrophobic pocket, and therefore they would rather leave, but they've got trapped. Okay, so the energy of EGFR APO is on the positive side, means not favorable. Now, if you have to design a drug for a receptor like EGFR, and this is just an example, which has trapped unhappy waters, there's an opportunity. You need to identify a ligand, okay, which has some hydrogen bond forming groups, and it can form some interactions, happy interactions with water. If this is the case, if you form the complex, once this ligand is part of the receptor, it will make interactions. So when it was in the apo form, the waters had no interactions. When the ligand comes in, the ligand is now making some of the waters happy, lowering its energy because it is able to form some hydrogen bonds. So what would happen? This will now make the complex energy much more stable than the empty EGFR. So the ligand itself has got, when it is solvated, its energy is on the lower side. But when it goes inside, when the complex is formed, the bound P plus L, the protein plus ligand, the energy of the protein, the waters, and the ligand goes much more on the negative side, and you get a boost in binding affinity. So this is one of the ways we can use waters for improving the binding affinity of a molecule to a target receptor. How do we know happy waters and unhappy waters? It all boils down to thermodynamics. Okay. So if the free energy, when we say happy, we are talking about energy being more negative. If the free energy of the water or its enthalpy and entropy components are negative, then it is a favorable water. If it is positive, it becomes an unfavorable water. So a software like WaterMap. Okay, so Schrodinger is one of the software companies who have the module of WaterMap. It's extremely costly to license out. So it's beyond most of us in academics, but it is there. So what they do is, when you have the ligand, the protein ligand complex, they computationally extract the ligand and they flush the active site with water. Alternatively, if you have the apoprotein, you can just flush the active site with water, and then the energy of interaction of each of these water molecules with the protein is computed. The energy has the free energy plus the enthalpy and entropy components. So you can see in this particular example, most of these waters which are the edge, okay, they are shown in golden or yellow, they have a negative energy, negative free energy. So they are the structured waters, or the happy waters. Whereas the pink ones, they are the ones with the positive energy, 0.7, 0.8, etc. So they are the disordered waters or the unfavorable waters. So if you have done the water mapping, now that if you want to design a ligand or try to see a ligand, what you can do is you can superimpose, or you can try to fit a molecule which has polar groups. So you can see here there's an NH, there's an NH2 in the positions where there were the happy waters. So what will happen when this molecule comes in? It will displace these waters out, but it will form the same interactions. It is mimicking the interactions of the happy waters. To add to that, in the place where there were the unhappy waters, the disordered waters, you can ensure that the molecule is hydrophobic. Okay, so once again, when this molecule binds, these unhappy waters will also be displaced out. But now, since you have placed a hydrophobic part of the ligand there, it is going to have a good binding affinity. If you have a polar group here, they will also be unhappy like the waters whom they are replacing. So this is the way one can use the energetics of the water map to design ligands with high potency. Okay, just one final example of a fantastic success story. HSP 90 is a target. the first candidate or the first lead molecule they got was this. It had very poor binding affinity, 790,000 nanomoles, almost millimolar potency. So they decided to use to see water map works. So this was HSP 90. Water mapping was the mapping of the energetics of all the water molecules were done. And then this molecule was, the complex was formed. Okay, so now you can see here that this ligand has displaced most of the waters. But in spite of that, there are some waters still remaining. The molecule was quite small. So you can see that there is this disordered water at this position, and there is this happy water here. So they used a very simple strategy. Wherever there were the disordered waters, they placed intentionally a methyl group. So this OCH3, this is OCH3, was replaced with an isopropyl. Okay. And since there is a happy water here, they placed an OH group here, which would displace this water out. So now what happens? You have made two small changes. You have replaced an OCH3 with an isopropyl group, and you have added an OH at this particular position. What will it do? This change will displace this unhappy water out. Okay. And you can imagine, those of you who are familiar with thermodynamics can imagine the tremendous improvement in entropy. This is a trapped water, unfavorable in the binding cavity. Isopropyl group is throwing it out. It's now in the aqueous media where it has freedom, entropic freedom. There will be a boost, a tremendous boost in the binding affinity of this molecule because of the entropy component of displacing an unhappy water. Secondly, you have put an OH here, which is going to make a hydrogen bond. Just like this water was making a hydrogen bond, this OH will also make a hydrogen bond and gain enthalpy. When there is bonding, it is enthalpy. So two minor changes, one improving the entropy, one improving the enthalpy. Binding affinity goes from 790,000 to 56 nanomolar. Okay, so that is the power of using, looking at the whole picture and actually trying to understand how you can boost the binding affinity, not just doing docking between the protein and the ligand, but also looking at the other parts, that is in this case, the waters. That brings me to the end of my talk. I have touched upon some SBDD, some LBDD, and some water map based techniques that are used to accelerate drug discovery and make it more rational. I have tried to give examples which highlight the power of these methods in lead identification and lead optimization, and hopefully given you all a peek into this tantalizing three-dimensional world of computational drug design. That's all from me. Thank you very much for your attention. If anybody has any questions, I'll be happy to take them.
Thank you, ma'am. We are having some questions from participants, ma'am. The very first one comes from our dear Vandana, ma'am. That is there any limitation with respect to molecular weight in softwares?
No. So if you are doing docking, there is no real limitation, unless if your target has a large pocket, you can also dock larger molecules. Okay, so yeah, so I'm probably getting, so if you're talking about say phytochemicals with a large molecular weight, some softwares may not be able to handle a large molecular weight. But if a large molecular weight molecule has less rotatable bonds, it's more rigid, then if you're doing docking, for example, it doesn't have to search that much. But if you have a large molecular weight compound with a lot of rotatable bonds as well, then the software does need to do a thorough or an exhaustive search to come or converge upon the bioactive. So it will change from technique to technique and case to case. Vandana, did you have some specific example in mind?
Yeah, in fact, I've written in the question itself, can I check activity of polymers as therapeutic moiety, because generally used softwares do not permit. And as you mentioned, receptor is huge and the polymer is also huge.
Yeah, so then there are, again, like Schrodinger has come up with material science. So there is this advanced technique of coarse graining. Okay, so when we use the regular life sciences protocols, we do docking of atoms. Individual atoms are docked where you have gone deep into the molecule. But if you have a large polymer, you can build the model of the polymer as groups. You are graining the model. You will not get finer insights, but you're able to complete the job. So coarse graining is used for larger molecules like polymers. And in fact, even things like polymers or cyclodextrins for seeing the solubility enhancement, there is this technique of coarse graining which can be used. So the force field is a little bit different. The energy equations are a little bit different because we are grouping CH3 together or CH2 CH3 together. So there is a system, a science for building a coarse grain model. But it is definitely available if you have the right software.
So Schrodinger has a module.
Schrodinger has material science, which does coarse graining of large molecules. So they have a complete, so the material science is able to build formulations. So you can actually say embed your drugs in chitosans, build your manos, and do all of those kind of studies. So they have developed their science. That's why they are so expensive because they are working on it. Their application scientists are updating the science at every point to give us what we need.
Thank you.
Thank you, ma'am. Ma'am, another question is there any minimum limit for docking score value or scoring function?
Minimum value?
Minimum value for docking score value or scoring function.
Again, it will differ from software to software. If you're using something like Schrodinger, so basically what I would suggest is the native ligand, you dock and see. The native ligand, if you're able to replicate the position, see the score of that. If it is -12, then try to design your analogs which are in that range. If the native ligand is giving you a score of say -9, you have used say Autodoc Vina, you're getting a score of -8 or -9. You will never get -14. Then that is the bar. So it will differ from software to software and from protein to protein again. But the more negative it is, the better it is. If you get a score of -4 or -5, even if your native ligand gets -4 or -5, that means you have failed somewhere. Your protocol needs to be optimized further. I hope that answers the question.
Yes, ma'am. Ma'am, another question is what changes need to be done to check MD of a salt or base form of a drug?
What can you repeat the question?
What changes need to be done to check MD of a salt or base form of a drug?
So if you are doing the MD of a charged form, that's how I understand it, like a charged form, you need to add counter ions. So any MD program like Desmond or Gromax, which are commonly used for doing MD, they will build your system. If it has some charge, they will neutralize it with counter ions. Okay, so sodium ions or chloride ions are added because you need to have you should have a non-integer charge on your system before the software can evolve it or perturb it. So addition of counter ions is done naturally by any algorithm which does MD.
Okay, thank you, ma'am. There are some more questions from Bombay College of Pharmacy itself. That in what extent we can rely on freely available docking software?
Autodoc Vina are accepted by publications. So let me not say with software, if you are able to replicate the bioactive pose, if you are able to match the SAR. So again, whenever you do docking, whenever you use any model, initially try to see if your predictive model is able to predict the experimental results. So if you're doing docking, for example, you should start with a training set where you have the activity values. If your docking protocol is able to replicate the SAR, highly active, more negative scores, low active, less scores. That means your protocol is able to manage. Autodoc Vina, validated, accepted by publications. So most of us who cannot afford a Schrodinger or an MOE, we do use Autodoc Vina. They also have a high throughput module called Pyrex for doing virtual staining, docking of large number of molecules. So there's some amount of user friendliness in that as well. But you need to have something like PyMol or Discovery Studio Visualizer to see to analyze the binding pool, because in that Autodoc tools, it's not that intuitive. You really have to like struggle with it. So these docking softwares are accepted.
Okay, ma'am. Thank you. Thank you very much for covering SBDD, LBDD, and active voter sites. And these are very useful for the drug designing. Actually, we are also from chemistry branch, and we need all this for the drug discovery and research oriented in our lab, ma'am. Other topics, there are also some vast scope in this field also. So in future, we are looking forward for your help also.
Definitely.
Yes, ma'am. Yes, ma'am. I think Dr. Somani has been joined.
Okay. Dr. Somani, will you like to say something on this while concluding today's sessions?
I think he has left.
He was not in the proper range, actually.
Yes. Okay.
So thanks,
Okay.
Dr. Aarti, for elucidating the things simply. And Vandana ma'am, we are always grateful to you for always being there with us and easing our learning process. So we can conclude today's sessions, and all the participants, please join tomorrow, same time, 10 o'clock exactly. And we will start with our sessions. Tomorrow is Dr. Ruxana Rab in the first session, and second session by Dr. Dhaneshwar. It changed as per the request of Dr. Indupal ma'am. So tomorrow there are sessions by Dr. Ruxana Rab and Dr. Sunila Dhaneshwar. So see you all then. Thanks everyone.
Okay. Thank you once again. Thank you.
चला जेवण करू. काय एक्झाम आहे नंतर त्याच्यामुळे म्हणलं सुरू करूया ना सुरू नंतर एक्झाम आहे 1 वाजता. डबा नाही या शुभम सर कुठं बसायचं इकडं काय इकडं बसायचं. बोलायला का आता काही? सर झालं ना सेशन, लिहू का की
फीडबॅक
तिथं आलंय ना त्याच्यावर झालंय ना.
कुठं आलंय?
कमेंट मध्ये, चॅट बॉक्स मध्ये.
नाही जॉईनच केले मी, मला फक्त लिंक
व्हॉट्सअपवर टाकले. मला काय वाटलं ते ह्याचे जो हे आलेत की हा अयो हे काय वेगळंच काहीतरी पेज झालं, सॉरी. पाठवली बघा. हे काय आलं एवढं मग कॉपी केलेलं. इस घटना से देश में कोई आग नहीं लगी. कशा आपल्या ऑर्डर असिस्टंटच्या पैकी सगळ्याची ऑर्डर असिस्टंटच्या पीसीआय साठी लागते ना असिस्टंट. आली का? गावात आणून दिली इन्स्टॉलेशन. उद्या म्हणताय का? इन्स्टॉलेशन मध्ये काय ते फक्त हे करून दे. शिवकन्या मॅडम सुट्टीवर आहेत. आत्ता मेसेज वॉल माउंट करून देईल. आणलंय का त्यांनी ते?
हॅलो यायले 5 मिनिट थांब.
वॉल माउंट आणून दे मग.
ते सोबतच घेऊन
हो ना, नाही त्याला तरी फोन आला ते त्याला म्हणायचं घेऊन ये. काय 200-300 रुपयाचा असेल ते वॉल माउंट करून टाका. ते इन्स्टॉलेशन म्हणजे सगळं सेटिंग बिटिंग थोडस सुरू करून देते. डिश ऑलरेडी आहे ना.
नाही तरी
सर सुनील सर तिथं जेवायचं माहिती बाहेर जेवायत का? जेवायचं तिकडं का म्हणलं? त्रास काय देणार? त्रास काय देणार?
बडबड
बसूया. बस तुला मावतो सगळेजण हळूहळू. तुला लागलं काय केलंय? इंटरनल आणि एक्सटर्नल दोन्ही एक्झामिनरसाठी ऑफिस ऑर्डर बनवली. तुम्हाला एकदा दाखवतो. नोटीस काढली आपण. इंटरनल एक्झामिनर एक्सटर्नलची सगळी कंबाईन नोटीस पण काढली. पण इंडिव्हिज्युअल नावाने ऑफिस ऑर्डर आपण काढत नाही. आपण कंबाईन ह्याच्यात बनवलंय. इंटरनल कोण, सब्जेक्ट एक्सपर्ट कोण, एक्सटर्नल कोण, त्याचं टाइम टेबल बनवून सगळं टाकले. प्रत्येकाच्या नावाने इंटरनल एक्सटर्नलच्या नावाने ऑफिस ऑर्डर काढले. विद्यापीठाने म्हणजे MSPT आम्हाला फक्त मेल पाठवलाय, त्याच्यात नाव आहे.
नाव आहे
MBA च्या वेळेस काय मी एक नॉर्म्स वाचले होते की त्याच्यात ऑर्डर लागतो म्हणून आपण काढले. मी तुम्हाला फॉर्मॅट दाखवतोय ना, बनवलाय जुन्या कॉलेजला आम्ही बनवलेला तोच आहे, तुम्हाला दाखवतो.
सगळ्यांचे नाव होते सब्जेक्ट वाईज. मी बघितलं होतं, मेल आलेलं आहे.
ऑफिस ऑर्डर म्हणून
एक परीक्षा अशी एक तशी
अॅन्युअलच्या पेपरला आपण ऑफिस ऑर्डरच काढलं सगळ्या. मी तुम्हाला हे सगळं फॉर्मॅट तुम्ही एकदा बघून घ्या. कारण की MBA च्या वेळेस ना त्यांनी असं ऑफिस ऑर्डर मीटिंग पण दाखवायचे रजिस्टर. एक
नक्की सांगतो ना, कालची भांडणाले पाहिजे
एक एक्झाम आहे ना,
शेवटची कोणाकोणाची झाली? विराट कोहली काय माहित नाही काय झालं, बाहेर आलं झाले त्यांचे मॅच संपल्यानंतर झालं. काय झालं ते मागच्या वेळेस गंभीर हे झालं होतं ना, बँगलोरला चिन्ह समोर त्यांनी गंभीर RCB बनवलेले, RCB हरले म्हणून गंभीर नाही का. आणि यावेळेस आता त्यांची लग्न हरले की हे नाही याला म्हणजे वाटलं नवीन
नाही सर देतील सरांकडून
चालवतात फोन चालले वाटाय
टेन्शन नाही. पण घेऊ पण तिने नाही कॅन्सल केली. कॉलेजात सांगितलं मी येत नाही म्हणून. काय कसं चालू आहे
काय स्पेशल काय आमची एक्झाम
आता 3 दिवस मग तुम्ही 2 घेण्याची ऐवजी आता गेले असते. नाही
हॅलो इथं मी ऑफिसला कुठे
सगळ्यात बाहेर मोबाईल मध्ये बोलू मध्ये
पडला म्हणून नाही करत मी बघितले ना आता दिसते बदडे आले ते आता काय तुटलाय एकदम छान दिसतो ना दिसायला तुटला गेला होते पण कसं एकदम छोटा आहे टाकून आपण गरज काय ओके ओके ह्याच कलरची बॉल टाकणार जातेच किती सर 700 रुपये तसं नाही म्हणत त्या कवरनी कुठे काय करते बॉडी कवर छान आहे नाही दिसायला कवर असं झाल्यावर दिसलं बघूनच टाकायला पाहिजे नाही, काढ नको. थांब ना, वरती cover केलंय, sir.